Here are 10 Puerto Rico Medical Board-style clinical vignettes focused on cellular biology structure, with 4 answer choices and detailed explanations. Vignette 1. Nuclear transport. A 32-year-old researcher is studying nuclear proteins. She notes that some proteins must be transported into the nucleus via a nuclear pore complex. What sequence is essential for this transport? A. Signal recognition particle. B. Nuclear localization signal. C. TATA box. D. Zinc finger domain. Nuclear localization signal. Explanation. The nuclear localization signal, NLS, is a sequence of amino acids that tags a protein for import into the nucleus by nuclear pore complexes. Without an NLS, large proteins cannot enter the nucleus. Vignette 2. Rough endoplasmic reticulum. A mutation affects protein synthesis in the rough endoplasmic reticulum. Which of the following cell types would be most affected? A. Neurons. B. Skeletal muscle cells. C. Plasma cells. D. Red blood cells. Answer: plasma cells. Explanation: plasma cells secrete large amounts of antibodies, which are proteins synthesized in the RER. Damage to the RER significantly affects these cells. Vignette 3. Mitochondria. A researcher discovers that a mutation in mitochondrial DNA impairs ATP production. Which of the following tissues would be most affected? A. Cartilage. B. Epidermis. C. Cardiac muscle. D. Bone marrow. Answer: C. Cardiac muscle. Explanation: cardiac muscle has high energy demands and relies heavily on mitochondrial ATP production. Dysfunction here leads to cardiomyopathy. Vignette 4. Cytoskeleton. A drug disrupts microtubule polymerization. Which of the following processes will be most directly impaired? A. Phagocytosis. B. Cytokinesis. C. Protein transcription. D. Intracellular vesicle transport. D. Intracellular vesicle transport. Explanation: microtubules are essential for transporting organelles and vesicles inside the cell, especially in neurons and dividing cells. Vignette 5. Lysosomes. A child presents with hepatosplenomegaly and developmental delay. Lysosomal enzyme deficiency is suspected. What is the likely pathophysiologic mechanism? A. Impaired exocytosis. B. Defective protein synthesis. C. Accumulation of undegraded substrates. D. Decreased ATP production. Answer: accumulation of undegraded substrates. Explanation: lysosomal storage diseases result from enzyme deficiencies that prevent breakdown of certain macromolecules, causing accumulation and cellular dysfunction. Vignette 6. Ribosomes. A newly developed antibiotic inhibits bacterial 70S ribosomes. What is the main effect of this medication? A. Prevents DNA replication. B. Inhibits protein synthesis. C. Destroys bacterial membranes. D. Blocks ATP synthesis. Answer: B. Inhibits protein synthesis. Explanation: bacterial ribosomes 70S differ from eukaryotic ribosomes 80S. Antibiotics like tetracycline target these, halting bacterial protein synthesis. Vignette 7. Peroxisomes. A patient with neurologic dysfunction has impaired very long-chain fatty acid metabolism. Which organelle is most likely dysfunctional? A. Mitochondria. B. Peroxisome. C. Golgi apparatus. D. Smooth ER. Answer: B. Peroxisome. Explanation: peroxisomes break down very long-chain fatty acids. Dysfunction leads to accumulation, contributing to disorders like Zellweger syndrome. Vignette 8. Golgi apparatus. A researcher studying protein processing observes failure to add mannose 6-phosphate to enzymes. Which organelle is malfunctioning? A. Golgi apparatus. B. Lysosome. C. Endoplasmic reticulum. D. Peroxisome. Golgi apparatus. Explanation: mannose 6-phosphate tagging occurs in the Golgi and targets enzymes to lysosomes. Defects can result in eye cell disease. Vignette 9. Smooth ER. A toxin inhibits cytochrome P450 enzymes. Which organelle does it primarily affect? A. Rough ER. B. Golgi apparatus. C. D. Mitochondria. C. Smooth ER. The smooth ER is involved in detoxification and lipid synthesis. Cytochrome P450 enzymes are located here. Vignette 10. Plasma membrane. A researcher alters the phospholipid bilayer to study membrane permeability. Which property of the hydrophobic core is crucial? A. Cholesterol concentration. B. Phosphate head group charge. C. Fatty acid saturation. D. Ribosomal protein content, fat saturation. Explanation: the hydrophobic core of the membrane consists of fatty acid tails. Saturation affects fluidity and permeability. Would you like another 10 questions on cell signal?